Hello again. We are going to go through part 4 of motifs and motif discovery today. This is part 4 of 5 parts. This is a short one connecting us from entropy to information. Again, these slides are available on Google Drive, and there is one activity associated with this. That activity, as always, is available as Activity 20 on eCampus. So let's talk about information content. Start by a little review from the last lecture. We calculated the maximum entropy, assuming that all states in a system are equally likely. And one of the more intuitive ways to consider this is as a roll of a die. If we roll a 4-sided die, for example, we have no ability to predict with any certainty what number is going to come up. And so we say all 4 of those states are equally likely. We can calculate a probability. That probability is simply 1 over 4 or 25% or 0.25 here. And we can calculate the maximum entropy by calculating the entropy in that system. Using this equation here, that turns out to reduce to log base 2 of t, where t is the number of states. So we can quickly calculate the maximum possible entropy in a system by plugging it into log base 2 of t given t states. Now, how do we use this information when we're talking about motif prediction for multiple sequence alignments? So first thing to note is that all possible states in a multiple sequence alignment are not equally likely. We know that by looking back to the frequency matrices or position-specific matrices that we calculate from a multiple sequence alignment. Here's our example. This is going back, I think, 2 sets of slides. So here are the different states. These are amino acid residues. Here are the positions in our alignment. And these are the frequencies that those states occur, that those amino acid residues occur in this position. So position 1, you can see leucine is there 100% of the time, so it has a probability or a frequency of 1, whereas all the other states are 0. Any states, any residues that don't appear in our multiple sequence alignment at all in any of the positions, we assume that their frequencies are 0. So we don't have all 20 amino acids in our matrix. We can reduce that matrix to just the states, just the residues that are included in our alignment. And that's why you only see a handful of states here. So how do we use maximum entropy? Well, first we start with the totally random model. We assume in our totally random model that all states are equally likely. We know how to calculate that up here. And then we say that the information at a particular position is the decrease in uncertainty from that totally random model. Let's put it another way. In this, we are assuming that information is a decrease in uncertainty. That's the basic premise of this calculation that we're going to go through today. Information is a decrease in uncertainty. Let's take an example. This position 1. The uncertainty in this position is 0. Based on the information we have, there is no uncertainty. We are totally certain that this position contains a leucine. That means the information we have here is at its highest level. We have the most information. It is a leucine. There is no wiggle room. Contrast that with position 3. We have some information. For example, we know there's no G, there's no L, there's no P, there's no T, et cetera, et cetera. We know that N occurs more frequently than the other bases or the other residues, but we don't have 100% certainty. So there is still some uncertainty. So the information we have in this position will be less than the information we have here. So higher uncertainty, less information. How do we calculate that? Information here as I, and that's just at any position. We're going to take the maximum possible uncertainty and we're going to subtract the uncertainty at that particular position. Now we know how to get this value here. What we're going to go through is how we calculate uncertainty at a particular position in our multiple sequence alignment. Using this equation, we can calculate the information content at each position. And to connect it back to motifs, positions with high information are well-conserved positions. They're positions that we might include in a motif. Remember, a motif is a conserved pattern. We can also think of a motif as a pattern with high information. So we know how to calculate max uncertainty. We need to know how to calculate uncertainty at a particular position. It looks like this. It's the same equation that we use for maximum uncertainty. But of course, these probabilities are now going to change because we don't necessarily have 4 or t equally distributed states. So to calculate information at a particular position, we're going to use this equation, probability times the log base 2 of that probability. And we're going to estimate probability using the frequency of a particular outcome. And in amino acid and nucleotide alignments, that outcome is the residue or the base. So we're going to use the frequency of particular bases or residues as an estimate of the probability of finding that residue or base in multiple sequence alignment. Okay, let's get rid of the easy ones first. Here's our hexapeptide motif. 5 out of the 6 positions are 100% conserved. Those are the blue ones. We can calculate the information in those positions. It looks like this. Log base 2 of 20, that's 20 states, 20 amino acids possible. That's the maximum uncertainty. Minus 1 times the log base 2 of 1. This is the only frequency, the only non-zero frequency in this position-specific matrix. If you need to, you can go back to the second slide in this lecture and look at that matrix. So that's log base 2 of 20. Log base 2 of 1 is 0. 1 times 0 is 0, of course. So this log base 2 of 20 minus 0 is 4.322. That's the amount of information in that position. That is the most information we can have. Now the real question is, how do we calculate information content when there's variability? When we have some amount of information, but it's not the most information. Again, we use our frequency matrix. So in the easiest situation, there is only one residue. We take a frequency of 1 times the log base 2 of 1. That turns out to be 0. That's the only calculation we need to make. In position 3, we have several possible outcomes here. They have different frequencies or different probabilities. In order to calculate the uncertainty at that position, we have to add up all of those frequencies times the log of them. So here it is written out down here. We have one outcome, probability of 0.7 times log base 2 of the probability. Let's say that's the outcome E. Here's the outcome for K. Here's the outcome for Q. Here's the outcome for V. Note that these are all the same because the frequency is all 0.17. And then we get to the frequency of N, the frequency of the N residue here. That's a little higher at 0.33 times log base 2 of 0.33. You do this math, you get this number in orange, 2.266. You take the maximum entropy minus the entropy at that position to get the amount of information, 2.056 bits. Now notice, roughly 2 bits is less than the 4.3 bits at the blue positions. Intuitively, this makes sense. The blue positions are 100% conserved. We have the maximum information at those. At this third position, we have less information, more uncertainty. Okay, a little exercise to help you understand this with some hands-on fun. This is activity 20. This is the last activity for the semester. As usual, it's up on eCampus. Part A of activity 20. What is the maximum uncertainty for any position in a nucleotide alignment? We've gone through this in a couple of different slides. Should be straightforward for you to calculate. What is the minimum entropy? Part B, I want you to calculate the total information content at each of the 8 positions in this alignment here. Total information content, that's what we just calculated here for a single position in this amino acid frequency matrix. I'm giving you a DNA alignment and asking you to calculate that total information content. To help you get started, I've provided an Excel spreadsheet. BIOL430 Act 20 XLSX. This is in the supplements folder on Google Drive. You may use this spreadsheet, fill in the blanks, do the calculations, and turn that in as your exercise, as your activity 20 answer. And then part C, what motif is present in B? After calculating the information content for each position in this part, you should be able to identify a region, a stretch of higher information. That's what a motif is. It's a stretch of higher information than the surrounding stretches. So part C, I ask you to identify the motif that's present in B. When you're done, submit that as activity 20. Okay, that's the end of this section. We'll finish up with part 5 in the next video. Thank you.